Hey everyone, this is the Scrutinizer, aka Dr. Meenakshi Noel. So I bring an interesting topic today, and I have named it as putting an end to Ivermectin saga for COVID. And at the same time, I want to give you a couple of simple tools that you can use to confirm if the information being given to you is in fact true or is it misleading and promoting a false hope. Saying this almost feels surreal that there would be an active spread of misinformation and a deliberate sale of supplements, drugs, and especially like ivermectin and putting lives at stake. I keep thinking, has the humanity really stooped so low that human life is worth nothing in this 21st century? I mean, where we boast of being an advanced society, that we can go to space, eliminate disease, alter genomes. On the other hand, there are people, they will sell their morals and ethics for what? Money. And is this money really worth more than another person's life? Or they do it because of their ego or maybe millisecond of fame? I don't know. But it is something to ponder about. What I do know is what has most worth in all eternity. It is a good scientific research done diligently that saves lives. Compassion on part of clinicians for the sick and dying patients, such that they don't do subpar clinical trials to get a publication with flawed data. And lately, this includes some researchers also, as I can see from ivermectin published literature. And now I won't waste my time or your time addressing the pharma companies because it seems they are so lost they won't even understand, and for whom it's only about making profit at any cost. So, having said this, to bring your attention towards the times we are living in, all of you need to start thinking seriously before believing in what you hear in the news or read on social media. And very sadly, I have to say that even the peer reviewed publications can't be trusted blindly, unlike the past where they were the gold standards. So, these are the tools I want to provide today, taking ivermectin as an example. And you should always remember the science is about facts, it's not about beliefs, loyalties, friendships, but it's all about facts, and the facts pass the test of time. And as usual, I don't want you to take my word for it. I would like you to look at the facts you already have for ivermectin, preferably not from Dr. Google, and compare with what I discuss here and use your critical thinking skills to make a good decision in your favor based on sound science. But no, you don't need to have any scientific background. I'm sure you know that all the videos I make and the articles I write are for general public, especially for the non-scientists. My goal is to stand up against misinformation and provide unbiased scientific reports to empower you so that you can ask the right questions and stop feeling helpless, and that you can quit being a blind follower. Of course, you can always take my help, write in the comments, and we can discuss or message me. And the good thing is, there is no cost involved. And this reminds me of a quote from one of my most favorite scientists, that's Einstein. He had said, if I had an hour to solve a problem and my life depended on the solution. So, see, very relevant to us for COVID, and our lives depend on the solution. So, he said, if I had only 1 hour to solve a problem and my life depended on the solution, I would spend the first 55 minutes determining the proper question to ask. For once I know the proper question, I could solve the problem in less than 5 minutes. So, what have we been doing to solve our problem in the past 3 years? We are recklessly taking shortcuts, being attached to our opinions, thus running away from the truth and shooting arrows in the dark and hoping. And what Einstein means by spending the first 55 minutes, meaning he would spend most of the time in figuring out what is the correct question, and then you can answer the problem. if you're ready now to get informed and use your gray matter for this discussion, let us all prepare to be dazzled with the shiny data that was robbing poor ivermectin of its good name. It's true that ivermectin has been a great discovery, but as an antiparasitic agent both in animals and humans, and rightfully, Doctors Campbell and Omura won the much deserved Nobel Prize in Medicine. Ivermectin has had an immeasurably beneficial impact in improving the lives and welfare of billions of people throughout the world by controlling nematodes, that is, roundworms, arthropod parasites, insects, etc. So, because of ivermectin, the world will soon be rid of two of its most disfiguring and devastating diseases. That is, for example, river blindness and lymphatic filariasis. So, thus, it has justly earned the title of wonder drug. And before this, the only two other drugs um, had the same title, that was penicillin and aspirin. I would like to point out that this is called doing the thorough ethical research to save lives, unlike what we see today. Okay, giving the name of a wonder drug, this does not mean that ivermectin can be used against all other pathogens like COVID-19 or bacteria, fungus, everything and anything. The key word here is antiparasitic and the dosage. So I will walk you systematically through the steps involved. The first thing to understand here is that any medication we make or discover is targeted solely towards the pathogen we want to get rid of, with minimum or no damage to the host. In this case, it's us humans. So, in a way, it's like finding the Achilles heel of the pathogen and targeting it selectively. So to clarify this point, I want to take a common example that all of us are familiar with. So let's take the example of sore throat, especially the strep throat. So strep, it's the nickname that we have named the bacteria that causes this throat infection. The full name is Streptococcus. Okay, so what is the treatment for this condition? You already know it's the antibiotics, but which one? So here, again, asking the right questions. Once again, we cannot just use any antibiotic. We need the one that will kill this particular bacteria. So amoxicillin or penicillin, they are in the same class of antibiotics, right? So how does penicillin class antibiotics cure the strep throat? In other words, how do they kill this particular bacteria? So remember, I just said we look for the Achilles heel of the bacteria, and in this case, that are round in shape, and that's why they are called the cocci. And we know bacteria is a single celled organism. So that is one round cell per bacteria, and they're grouped on the basis of their cell structure. We can only see it under a microscope, and this is what this picture is showing. So they are enclosed in a cell wall, which is very critical for the survival of the bacteria. So it protects the cellular structure that aids in their life processes. It gives shape to the cells, guards what can go in and out of the cell, and prevents them from bursting or lysis. So the strep bacteria will die if they don't have a cell wall, meaning this is a true Achilles heel of this bacteria. So the important step is to make an antibiotic that will inhibit the cell wall formation in this bacteria. And this is what the penicillin class of antibiotics do. So we know that these bacteria or any bacteria, they have to multiply in the infected person. So as you take this antibiotic, it goes straight to the bacteria and inhibits the cell wall formation in Streptococcus, which means the cell cannot be intact, and that leads to the death of the bacteria. So once again, like it said for ivermectin, this antibiotic has been safely used for years also. And the difference is that every strep throat does get better with it. Not sometimes or not in some people. It's without fail, it has treated the strep throat. So, this is what a good working functional medication means. Okay. So, why don't we use this antibiotic against everything else, like viruses, parasites, or COVID? As I just explained, it acts on the cell wall of bacteria, and viruses have no cell wall. Rather, they have a glycoprotein and envelope, meaning a sugar and a protein envelope around it. So even the parasites, they have a thin membrane called pellicle, but they don't have a cell wall. So it would be futile to use this drug for any pathogen that has no cell wall, because this antibiotic only targets the cell wall, and especially the cell wall that streptococcus, um, the constituents of that bacteria. So it would be futile to use this drug for any pathogen that has no cell wall, because this antibiotic only targets the cell wall, and especially a cell wall containing the same constituents as the streptococcus bacteria. So now you know why or how different medications are specially designed for different diseases or pathogens. So let's move on to our real target, that is ivermectin and coronavirus. Now, asking the same question, how does ivermectin work on parasites? So, the mechanism, ivermectin stimulates in the parasites, it stimulates the release of neurotransmitters in the peripheral nervous system of invertebrates or the parasites in excessive quantities. So, that will activate specific ion channels, which lead to an influx of chloride ions through cell membrane. So now this causes hyperpolarization in the parasite. It leads to muscle paralysis of the gut of the parasite. And this leads to the death, again, of the parasite. So, but the question for you is, would this mechanism work for a virus or even a bacteria? I really want you to think about it, and let me know in the comments. We can discuss. This is one of the major tools I promised to give you earlier, and I gave you detailed explanation on how to use it. And now I'm asking you this question. You only have to apply and come to an answer. Otherwise, I will definitely provide the answer in the next video. So, the next step now, after all this, is to address the dosage of the medication. As an antiparasitic agent, ivermectin, it is used as 200 micrograms or zero point 0.2 milligrams per kilo body weight of humans. The safest dose that kills the parasite without any harm to the host, that is us. So, to give you an idea of dosage of ivermectin, the LD50 for humans is 2 milligrams per kilo body weight. So, remember, I said that we use zero point 0.2 milligram per kilo body weight, and this is 10 times higher. And LD50 means the dose at which half the population would die. This is always the first step, which is determined for any new compound, drug, or medicine, along with studying the side effects of the drug. So, finally, now we have covered all the basics, and we can discuss a peer reviewed publication that started this whole mania. And now, this is the tool number 2, where I want you to know how or what to look for a publication, and decide the information that you're looking at. So, the authors of this study published that a single dose of ivermectin inhibited CoV2 by 5,000 fold. Now, which without reading the paper, it would seem like an exceptional news, and that's what we all have been waiting for. So their studies are done in vitro. What it means is that the cells are taken in petri plate, and in the incubator. So here they have the luxury of adding ivermectin until those cells die, because there is no organ system involved to study the adverse effects. So 50% inhibition of COVID-19 by ivermectin in vitro has been found to require an estimated oral dose of 7 megs per kilo body weight of the human, which is much higher than the LD50 of even the LD50 of ivermectin, which is 2 megs per kilo body weight. And this is high enough to be considered as ivermectin poisoning. So at high doses like these, ivermectin will easily cross the blood-brain barrier, and it will act on our neurotransmitters, activating our inhibitory chloride channels similar to those of the parasites, as I discussed above, and leading to the fatal consequence. So remember, we just discussed that the goal of any treatment is to keep the host safe and kill the pathogens. In this case, it is COVID-19. So not to kill the human, then of course, if we're killing the humans, then of course the CoV2 will die anyways, but that is not the goal. So why would this be a treatment for CoV2 patients if it is going to kill them? Think about it. Then lastly, the most important open secret of ivermectin and COVID-19. It's in biochem. I'm trying to simplify it, so listen it very carefully. So ivermectin is said to have antiviral activity. Sure, it does. But this is incomplete statement, and if I must quote, incomplete knowledge is very dangerous. So this statement that ivermectin has antiviral activity, it has led to confusion in both the patients and even clinicians as to why we aren't using ivermectin for COVID-19. However, the right question to ask again here would be, how much antiviral activity does ivermectin have? At what doses? And does it really inhibit COVID-19 in vivo, that is in the living system, or even in vitro? So the question again would be, how much antiviral activity does ivermectin have? Okay, number one, does it really inhibit COVID-19? And how does ivermectin exert its antiviral effect? So we have transport proteins. They are called the importin, alpha and beta 1. These are nuclear transport proteins. Now, these proteins are responsible for entry of enzyme called integrase into the nucleus of our cells. And there are many viruses, especially the DNA viruses. They rely on this important alpha and beta 1 dependent nuclear import for robust infection. So ivermectin's antiviral activity is due to its ability to bind this protein, the imp alpha. When ivermectin binds to it, this induces conformational change that prevents its normal function, which is helping in nuclear import of the key viral and host protein. So here is the open secret. COVID-19 is a it is a positive single-stranded RNA virus. It replicates in host cytoplasm. It does not target host nucleus. An entire antiviral activity of ivermectin is based on preventing the normal function of the nucleoprotein. And further, CoV2, it does not contain either integrase or NS5. It uses entirely different pathway to infect, and it involves either the ACE2, as we know, or the TMPRSS, and lately there was another mechanism, bradykinin pathway. So the question we all need to ask is, if the required enzyme is not even present in the virus, so how can we say that any antiviral or antiparasitic drug will act on it? So I really want you to ponder over it, that if COVID-19 or COV2, it does not have the required enzyme on which ivermectin will act and bind, and that's how it exerts its antiviral effect. And secondly, when our virus does not even go in the nucleus, as it's required for ivermectin to act upon. So this is what I want you to think about, and let me know by commenting below. And I will discuss a couple of publications in the next video, and they would be on ivermectin to, provide a proof. So if you enjoyed this information and you could like and subscribe to this channel so you can receive the notifications when I post another video. And like I said, the next video would be discussing a couple of published articles claiming usefulness of ivermectin for COVID-19. And I'm going to post the references below so you can even read those in advance if you want, so that it's easier to understand next time, but you don't have to. And finally, as I always say, think critically, stay healthy, the scrutinizer out.